How to predict success of vaccine without a big trial. Vaccines can trigger diverse immune responses, including the manufacture of antibody molecules that bind and block infections, viruses, and the activation of T cells that kill infected cells. Moderna's vaccines provides protection against COVID-19 by triggering the production of antibodies against a key viral protein. A study in monkeys suggests the insight, if confirmed in humans, could spread the development of next generation vaccines. Vaccines can trigger diverse immune responses, including the manufacture of antibody molecules that bind and block infectious viruses and the activation of T cells that kill virus infected cells. By identifying the immune responses that can predict a vaccine success, scientists could more easily judge candidate vaccines. A COVID-19 research update published on Tuesday by the scientific journal Nature said the update is part of a series of Nature summaries of key papers as they appear. To identify which immune responses are important for Moderna's vaccine, Barney Graham and Robert Surder at the US National Institute of Allergy and Infectious Disease in Bethesda, Maryland, and their colleagues gave monkey a range of vaccine doses and exposed the animals to SARS-CoV-2. Immune correlates of protein by mRNA 1273 immunization against SARS-CoV-2 infection in non-human primates. K.S. Corbett at Yale preprint at Biox. The vaccinated animals that had the lowest levels of viral genetic material in their noses and lungs also had the highest level of antibodies that recognizes the viral spike protein, the molecule that the Moderna's vaccine encodes. Levels of other immune markers did not correlate as strongly with the vaccine's protective effects. In one sentence summary of their findings, the researchers said mRNA 1273 vaccine induced antibody responses are a merchantistic correlate of protection against SARS-CoV-2 infection in NHP. mRNA 1273 is the technical name for the vaccine developed by Moderna. NHP stands for non-human primate, that is monkeys. The findings have not been peer-reviewed. A parallel study now underway will compare immune markers in people who were protected by jabs, including the Moderna's one with markers in people who were infected despite receiving a vaccine. Identifying these correlates of protection will help researchers to assess existing and future vaccines without running costly large-scale clinical trials. Thank you.